Hi, I'm Dr. Helen Smith. I'm faculty at the University of the Incarnate Words, Feik School of Pharmacy, and I'm giving you a presentation on some of the current updates on pharmacogenomic resources. This is what I'll be talking about today. We're going to just give a brief introduction about what pharmacogenomics is. Then I'm going to walk you through some of those current resources that I tell my students about, including a couple of databases, some guideline documents, and then a website that I think is really helpful. And then we'll briefly discuss the future directions of where pharmacogenomics is going and places where resources might become available. The objectives for this discussion are the following. The first one is, when we're done with this presentation, you should be able to list current pharmacogenomics resources. The second objective is that you will be able to compare the content and utility of these resources. And finally, you will be able to explain the weighting of evidence in one of the guidelines, the CPIC guidelines that I'm going to present. So let's get started with just our introduction on what pharmacogenomics is. By definition, pharmacogenomics is the study of how individuals' genetic makeup affects their response to medications. This is a field in medicine that combines pharmacology with genetics, and it's being used to help individualize patient therapy, which will optimize patient outcomes and minimize adverse effects. This field has been progressing very rapidly. It started in about the 1950s with the observation that there were inter-individual variation in drug responses, usually based on ethnicity or racial background of patients. At that same time, we started being able to identify some of the variants, the genetic variants of our metabolizing enzymes. And then after that, this field moved along, and the Human Genome Project was launched in the 1990s. And once that project was finished, we had a lot of tools at hand that we could now start investigating whether or not we could use information about genetic variants in individualizing medicine. So now we're at the present day where pharmacogenomics is being used in quite a few different practice areas. So the speed at which this has progressed is requiring that clinicians have available to them up-to-date resources. Along with the rapid speed that pharmacogenomics is progressing is the fact that we have many patients now being prescribed medications for which pharmacogenomics can have an impact. In 2023, a group led by McKinnon did a study where they evaluated patients' EMRs, and when looking at about 84,000 patients, they found that more than half of them were being prescribed medications for which we have pharmacogenomics information that can be clinically applicable. We also know that pharmacists and other clinicians, even though they agree that pharmacogenomics is important in clinical care, they're not always feeling comfortable or confident in their knowledge that they have of pharmacogenomics. And this was published in a recent study cited there on the slide by Wondrasek in 2024. Dive into a couple databases and guidelines and websites that you might find interesting as you're thinking about how do we use pharmacogenomics in patient practice. The first database that I'd like to get you into is the FDA's table of pharmacogenetic associations. On this slide, I have text from that FDA website, and it's saying that the FDA believes there is sufficient evidence that patients with certain genetic variants are likely to have altered drug metabolism and differential therapeutic effects and or risks of adverse effects. So they've created a table addressing this idea. Medications that are listed on these tables don't necessarily mean that the FDA at that time is advocating for pharmacogenetic testing to be done before patients are prescribed these medications. They're just letting people that are interested in knowing that there may be some pharmacogenomic associations with these drugs. As listed on the bottom of my slide, the table is not intended to affect current regulatory requirements or policies of these drugs, and it does not make an assessment of the safe or effective use of or the regulatory requirements for the detected variants in these genes that are referred to. And this table is not supposed to be used as a comprehensive information source. But let's look at this table. I've got a screenshot of the table in just a moment here, but I first want to tell you the drugs are listed in 3 separate sections in this table. The first section are medications that the FDA felt at the time they made this list that these were drugs that pharmacogenetic associations are supporting therapeutic management recommendations. The second section of this table is listing pharmacogenomic associations that could be causing potential impacts on safety or drug response. And then the third section of this table are medications that are listed that pharmacogenomic associations are demonstrating a potential impact on pharmacokinetic properties of these drugs. So this is a screenshot of that FDA website showing you the top of the first table or the first section. And I just wanted to show this to you so that you could see how the table is oriented. You've got the medications listed in the first column. The second column is listing the genes that these medications might be affected by. The third column is listing the variants or the phenotypes that have been identified that are associated with these medications. And then the last column is just a description of that gene-drug interaction. So these tables, these section 1 and section 2 and section 3 tables of these pharmacogenetic associations are interesting. I feel like they have a few limitations. I think that they're useful for helping people just to start looking at drug and pharmacogenomic associations. This table does not list any guidance for clinicians to use this information, and it has not been recently updated. But still, I think it's a really good starting point if you're just kind of getting into this topic to start seeing what do we mean by drug and gene associations. The next table that I want to talk about is one of my favorites. It's the FDA table of pharmacogenetic biomarkers in drug labeling. This is a table that I teach my pharmacy students about because it has lots of good information. This is a list of FDA-approved medications which have pharmacogenomic information in their labeling. It also will include specific actions that could be taken based on the biomarker information that is presented about this drug. It lets us know that the pharmacogenomic information can appear in various parts of a drug's labeling. And on the fourth bullet on the slide, you can see the biomarkers that are included. And then on the bottom bullet on this slide, you can see that this table does not include non-human genetic biomarkers. It does not include biomarkers that are used for just diagnostic purposes. And it is only talking about biomarkers that are related to the drug that is referenced and not to other medications. So here is a screenshot of the top of this FDA table's website. And this table is interactive. So you could, once you get into this website, you can sort the drugs alphabetically. And the drugs are listed in the first column. The second column is a listing of the therapeutic area that these medications are used. And again, you can sort that column and group these medications by their different therapeutic applications. The third column lists the genetic biomarker that is being associated with the medication. And then the far right column is information about where you can find pharmacogenomics information in the drug's labeling. So all 4 of these columns can be sorted, which makes it very helpful when you are making your way through this now pretty long column. Another way that you could access this table besides getting into this interactive table is that you could get into a PDF, a downloadable PDF of this table. And I know this is very small for you to see on these slides, but this is really the same information that I just showed you on the slide before. But on the far right column in the PDF version, you can see the actual extracts from the drug labeling that contains the pharmacogenomics information. So this is a quick way to get into the information that the FDA has on the drug labels, talking about what do these genetic variants listed here do to the effects of the drug or the risk for efficacy or risk for side effects. So I find this FDA table of pharmacogenomic biomarkers and drug labeling very helpful. It helps clinicians make or find information so they can make informed decisions about how they might change medication therapy based on pharmacogenomics. There's just a couple limitations with this table. It's only going to be listing drugs that are currently approved by the FDA, so any drugs in the works are not going to be listed on this table. And even though this table is updated quite frequently, it's not going to be listing emerging pharmacogenomic information that hasn't yet made it into the drug label. Another resource that I'd like to point out to you are the CPIC guidelines. This is the Consortium for Pharmacogenomics Information, and these are guidelines that are evidence-based, and they are to be used by clinicians to help understand how to use available genetic testing results from patients rather than whether to do genetic testing on patients that could be used for standard or individualizing their care. So these CPIC guidelines have been endorsed by several different organizations, and they're listed here on this slide. These guidelines are all written in the same type of format. You can see that there's always an introduction, then there's a discussion of the literature review process that went into making these guidelines. Then there'll be a discussion about the genes and the drugs that are being presented in this guideline. Any dosage recommendations that are suggested by these guidelines are then discussed, and then potential benefits and risks of using these guidelines are also discussed. And any caveats will always be put at the near the end of these guidelines. These guidelines are recently now including some tables of phenotype genotypes that you'll be seeing, and then when the information about prescribing is impacted by the genetic variant, you'll see tables following that will easily show the dosing recommendations that are presented by these guidelines. These CPIC guidelines go through a rigorous development and review process. They are developed by a group from the CPIC, and they do an initial assessment of determining which gene-drug pairs should be presented in the next guideline by doing a member survey and also surveying people that are stakeholders to this process. Once they decide which gene-drug pairs should be evaluated next, they form a writing committee, which is multidisciplinary, and it's also voluntary. And so this group gets together and they review the literature and they start drafting a guideline. Once they've drafted that guideline, all the evidence that was used to develop this guideline is weighted, and then therapeutic recommendations that are made in this guideline are also evaluated and weighted with their strengths listed using a transparent system that was borrowed from the NIH guidelines on using antiretroviral therapies. So the review and updating process for these guidelines is fairly frequent. They usually review these guidelines about every 2 years. It goes through a public and peer review updating process as well. So it takes quite a rigorous effort to get these guidelines published. I wanted to give you a little bit of information about the scoring of both the evidence and the strength of the clinical recommendations made in these guidelines. So the evidence that is used to make these guidelines is scored as either high, moderate, or weak. And then the recommendations are scored as either strong, moderate, or optional. So those strong recommendations are usually considered to have very high quality evidence supporting those recommendations, and the effects of the pharmacogenomic or the gene and drug relationship is usually pretty clear, and using those recommendations is considered to be fairly desirable. This is a current list of the CPIC guidelines available. I know this is quite small, but the first one The first one that was published came out in 2013, and we just had 2 published this last year. There was one that came out on methadone, and then another that came out on beta blockers. So we now have about 28 guidelines, 29 guidelines, and they're covering a number of different genetic markers in these guidelines. This is a screenshot of one of the guidelines. This is one of the newest ones. It came out in 2023. It was talking about the SSRIs and some of the variety of genotypes that we are finding are associated with response to these medications. So I'm not able to get onto the hyperlink to actually take you through this guideline, but these guidelines are rich in information. There's many citations that can lead you to the basic science that is used to support these recommendations. And I think they're a great resource for clinicians to use if you care to try to use pharmacogenomics in your practice. There are, the one limitation that I can think of about using these guidelines is that there are just a limited number of these out there so far. There are more in the works. It just sometimes takes a little while for our science to be available and to be of high enough quality that they can write the CPIC guidelines to use pharmacogenomics in clinical practice. So the CPIC guidelines are American guidelines. There are several other English language guidelines also out there. There's guidelines put out by the Dutch Pharmacogenetics Working Group. There's also a Canadian guideline called the Canadian Pharmacogenomics Network for Drug Safety. And then there's also the French National Network of Pharmacogenetics that has put out guidelines. All of these guidelines, including the CPIC or American guidelines, use similar methodologies for development. They do have slightly different objectives in their writing. So the Dutch and the CPIC guidelines, they focus on pharmacotherapeutic recommendations for drug and gene or drug and phenotype combinations. The Dutch and the Canadian and the French guidelines, they will recommend specific genetic testing to be done. The CPIC tends to stay away from recommending specific genetic tests to be used. And then the French guideline also will sometimes specify a clinical setting or a practice area that these guidelines could be used for. If you want quick access to these guidelines, including the CPIC, I suggest jumping on to a website called the PharmGKB website. You can see that name listed at the top left of this slide. And on the PharmGKB website, you can have access to these other guidelines besides the CPIC. So on this screenshot from the PharmGKB's website, I have shown you that if you look at the first drug, which is abacavir, you can see that there is the CPIC guideline, and then also the Dutch guideline is available for that medication. If you scroll on down through this web page presented by the PharmGKB, you'll see some medications have the Canadian guideline listed. Not every medication on this list has a CPIC, so the PharmGKB website is listing all guidelines available, whether they are the CPIC or not. So now I want to talk a little bit more about that PharmGKB website that we just looked at to see all the different guidelines that are out there. This is an NIH-funded resource, and it provides information about how human genetic variation affects response to medications. The PharmGKB collects and curates and disseminates knowledge about clinically actionable gene-drug associations and genotype-phenotype relationships. So I've listed on this slide some of the key features that are presented on this PharmGKB website. There's prescribing information, there's drug label annotations, there are clinical annotations from some of these guidelines, there are variant annotations coming from the literature, there are curated genetic pathways that are presented, and there are a genotype selection interface, which is a new feature of this website where you can get in there and try to get some recommendations for what genotyping you might be wanting to do. So this is a screenshot that is fairly current for the PharmGKB website, the front page of the website. this is a very busy, information-packed website. And as you can see on this website, there's lots of information, like I just talked about, that can be found on this website. I really like this website because you can dive deep into clinical recommendations. You can also dive deep into the science backing up these clinical recommendations by diving into the citations that the PharmGKB puts on this website. So those are the various pharmacogenomic resources that are currently available. I think they're some really good tools. There are a few directions that pharmacogenomics is going to be heading into. And of course, we're always needing to continue to educate our healthcare providers to improve our understanding of pharmacogenomics. We also need to be educating our patients more about the value of using pharmacogenomics when it's appropriate. We need to be expanding our list of genetic tests that we could do to help individualize our patients' care. But where resources might also be going include our ability to use pharmacogenomics in practice. So there's a need to create some clinical decision support systems in practice settings, and also the ability to integrate pharmacogenomics data into electronic health records. So those last two bullets here, the need to develop clinical decision support systems and our ability to put pharmacogenomics patients' information into EHRs, those efforts might start generating other resources that we will be tapping into as they become available. So I thank you for this time with this presentation. I am more than happy to answer any of your questions that you might have. So please feel free to shoot me an email if you want more information about these resources, and I can be found at hesmith@uiwtx.edu. Thank you.